Hello, AP Bio. Welcome to our video lecture on Chapter 6, An Introduction to Metabolism. So the picture I chose for this chapter is a picture of my son, Jack. He's probably maybe 2 in this picture, and he's pretty tightly holding on to that ear of corn, that piece of corn on the cob. And what you don't know is right before this picture was taken, either my wife or I had sort of reached for the corn to cut the corn off the cob, but Jack was concerned we were going to take it away from him like permanently, and he was not happy about it because he was very, very hungry and he was very possessive of his corn on the cob. And the look on his face is kind of saying, don't come near me or I might, like throw the corn at you or something. But as most 2-year-olds, Jack had a very healthy metabolism. He was a growing boy, which is why I picked this picture for the chapter. So let's dive right in. So this is a pretty important chapter. Your body is basically a collection of millions of chemical reactions happening all at the same time. Your body digests what you ate, your body builds the molecules that it needs, it fights off disease, your brain runs the whole show. And basically the cell takes the energy from the things that you eat and it uses the energy to do whatever it's going to do. This picture shows a bioluminescent squid, I believe. And you can see that the squid is converting the energy from whatever it ate into light energy, and that it's actually glowing. It's bioluminescing, it's giving off light. So the main sort of scheme here, so the definition of metabolism is just the totality of an organism's chemical reactions. And most reactions that happen in your body happen with the aid of an enzyme. And really for just about every reaction your body does, it has its own specific enzyme that catalyzes that reaction. So this picture is showing a sort of an enzymatic pathway where you start with molecule A, which molecule A might be something that you ate. And it turns into molecule B and then C and then D, which is what your body wants to produce. So in this case, B and C are intermediates on the way of producing D. And each of those reactions, there's 3 reactions that you need to get to D, is catalyzed by its own enzyme. So for instance, if you did not have enzyme number 2, say enzyme number 2 is coded for by a gene that you have a mutant version of, and enzyme 2 doesn't work, the levels of B would go up and the levels of C would go down. If you didn't have enzyme 3, the levels of C would go up and the levels of D would go down, all right? So catabolic and anabolic, so you can basically divide all the chemical reactions into your body into 2 categories. Catabolic reactions are ones that break things down. So when you ate your sandwich for lunch and your body digests that, those are catabolic reactions. Generally speaking, any reaction that breaks a bond releases energy, and the sort of notation for that is downhill, all right? Think of chemical bonds as being shared pairs of electrons, that's what they are. If you break a bond, it's like you're freeing that pair of electrons. You're not quite freeing it, but think of it as those electrons are available to do something, and that's going to be releasing energy. Building bonds, reactions that build bonds usually require energy, a net input of energy. Those are called uphill because they need energy to build the bond. Think of it as in building the bonds means you have to take electrons and put them together into a bond. That usually takes energy. And what your body does is it couples anabolic and catabolic processes together. So if I need to build a piece of DNA because the cell is going to divide, that's going to be a anabolic reaction, building something. So maybe the food that I ate for lunch, your body digests, that's catabolic, stores that energy is ATP, which we'll get to in a minute, and then uses the energy to build molecules. I think we defined these terms in chapter 2 or chapter 3, so we don't really need to go through them again. But potential versus chemical energy. So potential energy is just the energy based upon an object's location. This picture from your book shows a diver. The diver on the platform has lots of potential energy, but no kinetic if they're motionless. The diver halfway down, basically you converted the potential energy into kinetic energy. So let's say this individual has 10 joules of energy, joules the unit of energy. Halfway down, so say this was 10 joules of potential energy. Halfway down, you have, say, 5 joules of potential and 5 joules of kinetic because you're moving. When you hit the water, you would have 0 joules of potential and 10 joules of kinetic, all right? Energy can be converted from one form to another, but it cannot be created nor destroyed, with the exception of nuclear reactions, which we're not going to worry about in AP Bio. Thermodynamics is a topic that usually you study in chemistry and physics. We don't do much of it in biology, which is great because everyone usually dislikes thermodynamics. It's basically the study of transferring energy. So a closed versus an open system. A closed system would be one that's completely isolated, like having a beverage into a thermos that lets no heat escape. An open system is one where you can exchange matter, energy with the surroundings, which you are an open system because you eat, you breathe in oxygen, you breathe out CO2, you produce waste. You're an open system. You exchange nutrients and energy with your surroundings. It's very, very hard to have a true closed system. Even the Earth is really an open system because asteroids can hit the Earth. When we send people into space, that means we're an open system because the Earth is losing mass, technically. The two laws of thermodynamics we're going to discuss. The first law says the energy of the universe is constant. We said this in a previous slide. Energy can be transferred, so I can turn light energy into heat energy. If I have a very bright light bulb, some of that light gets turned into heat because the light bulb gets hot. On a warm summer day, if you wear a black shirt, the black shirt gets pretty hot because some of the sun's light energy is converted into heat energy. The second law is the law of entropy. Entropy is sort of a vague concept. What a physicist probably wouldn't call it vague, but us biologists call it vague. It's a measure of disorder. And in terms of our purposes, every time that you transfer energy from one type to another, some energy gets lost. All right. Energy being lost is another way of saying that you're getting more disordered. All right. When I drive a car, when you combust the fuel in the car, that's to make the car go, right? But the car gets hot. The hood of the car can get very hot because some, and actually most, of the energy of the combustible engine is actually lost as heat, right? When you digest a meal, that process isn't 100% efficient, where you're going to lose some energy as heat, which in this case would be your body temperature, which your body actually uses that for homeostasis. So there is a reason for the heat to be lost. It's not completely wasted. But you digesting that sandwich, your body doesn't convert all the energy into ATP. Some of it's lost. That's because of the second law of thermodynamics. This picture, this is a lovely picture, shows a bear eating a fish. And that's chemical energy, and the bear uses the energy to, in this case, move, but the bear is also giving off body heat. Some of the energy is wasted or is released as heat energy. Heat is considered the lowest grade of energy because heat, like when your body gives off heat, it's not storing the energy. It's hard to put heat to work. Using the energy of your lunch to make ATP puts the energy to work because you're going to use the ATP to do something. Wastefulness being released as heat, that's kind of cheap energy because it doesn't really do anything for your body. Your book has this great quote: organisms are islands of low entropy in an increasingly random universe. The entropy of the universe is increasing, which means the disorder of existence is going up. But you are not disordered, you are ordered. You take in oxygen, you take in carbs, and you build intricate structures like DNA or proteins or carbs or lipids. Building intricate structures out of atoms is decreasing entropy because you're making it more ordered. All right. So the cell, or even you as an organism, are basically like an island of low entropy because you're organized in a universe of increasing entropy. Of course, when you die, you're going to decay, and all the great orderedness of your cells will decompose. The entropy will eventually start going up again. These two images from the book just show very complex structures. The way that nature takes randomness and turns it into things that are organized is pretty beautiful, actually. So the concept of free energy, the textbook goes into way more detail into this. But back in the old days, we had to do some math with this and do delta H and delta S and lots of things that we don't need to do now. But you do need to know what delta G is. So delta G is the variable for what we call the change in the free energy. And what we're talking about is when a reaction happens. Does the reaction give off energy or does it require energy? If the delta G is negative, that means that it gives off energy. So when that squid was glowing, that reaction had a negative delta G. If the delta G is negative, it means the reaction is spontaneous, which means it can happen all by itself. It doesn't need any help at all. Help might be useful, but it doesn't need help. If the delta G is positive, it means the reaction needs energy to happen. You have to put currency into it. It's not spontaneous, okay? These graphs, so exergonic versus endergonic, you probably did this in chemistry. Exergonic reactions are ones that give off energy. So if you break a glow stick and it lights up, or you have those hand warmers in the winter when you crack the hand warmer and it gives off heat, those are exergonic. That means that the energy of my reactants was higher than the energy of the products because you gave off energy. So when you see a graph like this is telling you the delta G is negative because the energy of the system went down because you gave off energy. Lots of times kids get these confused. I'm not quite sure why. If the free energy, if the delta G goes down, it means delta G was negative. All right. And endergonic reaction is one that requires energy. So building DNA will not just spontaneously build by itself. You need ATP. You need a source of energy to build DNA. So my reactants would just be the atoms. My products would be a double helix of DNA. That took energy. All right. In this case, delta G is positive. It requires a net input of energy for it to happen. So exergonic is digesting your lunch. Endergonic is building a new cell. This picture from the book, so the way that a hydroelectric plant works is you're using water flowing downstream, which water flows downstream due to gravity, and you have a turbine here, and when the water turns the turbine turns that into electricity. Here, this shows a delta G that's negative because the water is going downhill, so I'm giving off energy. And if I can harness that, we can make electricity. Here, the water level is balanced, so delta G is 0, and the turbine obviously is not turning. To get the water to go back uphill, if you did that, that would take a net input of energy. That would be endergonic. That would be a positive delta G. What the cell tries to do, what your body tries to do, is to take this scenario, but break it into steps. So it's like you're being more efficient at harnessing the energy, in this case due to gravity. And at every single step, you have a smaller delta G, but it's still, or a smaller negative delta G, but delta G is still negative. You're just harnessing the energy at more points, so overall you're going to get more. So this brings us to ATP. So we first met ATP in chapter 3, I believe. Maybe it was 2. I think it was 3 though. So ATP is adenosine triphosphate. It's a molecule that has 3 phosphate groups. This is the ribose sugar, and this is adenine, which together you call this part adenosine. Remember, it's a nucleoside. If you cut out the phosphate groups, that's not that important of a word to know. And remember how we said these 3 phosphate groups, that's a -4 charge. It's not stable. So ATP would love to kick off that phosphate group and form ADP. This reaction right here shows the dephosphorylation of ATP. So I have ATP, I kick off the phosphate group. The symbol is a P with a circle and a subscript I, lowercase I, that stands for inorganic phosphate, and you gave off energy. This is called the hydrolysis. It also requires water. This is called the hydrolysis of ATP. This, ask yourself real quick, is this reaction endergonic or is it exergonic? Positive delta G or negative delta G? All right, think for a second before I tell you. All right, because this reaction gives off energy, it's exergonic, right? And because it gives off energy, the delta G is negative. Is this reaction spontaneous? Yes. All right. The reverse reaction would be opposite. The reverse reaction would be endergonic. It would have a positive delta G and it would not be spontaneous. Okay. So how does this work? So this number, -7.3 kilocalories per mole, if I have a mole, if I do this reaction, if I have a mole of ATP and I hydrolyze all of it, you produce 7.3 kilocalories, all right, for 1 mole. It's negative because you're giving it off, all right? So that's like saying I had a mole of ATP, I sold it. How much did I get? I got $7.30. And because you got it, it's negative. So that's $7.30 in my wallet that I can spend to do other things. Now, look at the reaction that's on the PowerPoint. So Taking glutamic acid, which is an amino acid, and adding an extra ammonia group forms a different amino acid called glutamine, all right? That reaction is not free. That reaction costs you 3.4 kilocalories for a mole, all right? This reaction has a positive delta G. It is not spontaneous. It is endergonic. But guess what? I need glutamine. It's one of the amino acids that you need to build proteins. So how do I do it? Well, this looks complicated, but it's actually not. This is the reaction that I want to do. All right, it costs me 3.4 kilocalories per mole. Well, breaking down ATP gives me 7.3 kilocalories per mole. You don't need to memorize that number. The AP exam would give you the number, but you need to know what it means. So do I have enough money to buy to buy a mole of glutamine? Well, if I have 1 mole of ATP, you do. And what happens is you go through what's called a phosphorylated intermediate. All right, this is a very important concept. The ATP, that third phosphate group, it gave it to the glutamine. Gave it to the glutamic acid, all right? Giving it to the glutamic acid increases its free energy, right? This bad boy right here will bond with the ammonia and be spontaneous to form glutamine, all right? So just do the math. This requires 3.4. Hydrolysizing ATP gives me 7.3. Do I have enough? I do, and you have some left over too. So if I have 1 mole of ATP, can I form 1 mole of glutamine? Yes, you can. And the way you do that is you phosphorylate one of the reactants, which increases its free energy that's available, all right? When ATP lost that phosphate group, it became more stable, it chilled out. ADP is a much more stable molecule, but it made the glutamic acid less stable. It increased its free energy, which the reaction obviously uses to make glutamine, okay? This is simple math. If I wanted to build something that costs me 10 kilocalories per mole, how many moles of glucose do I need? Well, you need more than 1 because 1 isn't enough. 1 just gives you 7.3. If I have 2 moles of ATP, can I make something that costs me 10 kilocalories per mole? Yes, you can, because you have 2 7.3s, you have 14.6. Okay, hopefully that makes sense. Breaking down ATP gives me cash. Building molecules costs me cash. Think of it that way. I just said this has ATP stored energy. So that phosphate group is kind of like if a bee is flying around you, you get higher energy, right? Because that bee, you don't want it, right? Well, the bee is sort of like that third phosphate group. If I phosphorylate the intermediate, phosphorylate one of the reactants, you're going to increase its energy and make the reaction spontaneous. Sort of like it's sort of like, I don't know, playing hot potato. Nobody wants that third phosphate group, so people keep passing it on. And when you get it, it increases your energy. ATP is a great molecule because you can regenerate it. So this is the hydrolysis of ATP. This arrow, if I ask you what was the delta G, and you don't need to memorize the number, but say this was multiple choice. This is negative 7.3 kilocalories per mole. So if that's negative 7.3, what does it cost me to build a mole of ATP? It costs me positive 7.3 kilocalories per mole. All right, switching gears real quick. So enzymes. Enzymes are catalysts. Catalysts are molecules that speed up reactions. Catalysts are great. They speed up a reaction without being consumed by the reaction. So if an enzyme, catalyzes a reaction over here, it can go on to catalyze a reaction somewhere else. It keeps working over and over and over again. Enzymes are called catalytic proteins. Not every catalyst is a protein, but enzymes are proteins, so you call them catalytic proteins. The picture that's on the board or on the PowerPoint here, so sucrose is a disaccharide, to remind you from chapter 3. Sucrose is a glucose covalently bound to a fructose, right? Your body can break down sucrose into glucose and fructose to give you energy, right? The enzyme that breaks down sucrose is called sucrase. Enzymes typically end in the suffix ase. So it's pretty easy. What enzyme breaks down lactose? Lactase. What enzyme breaks down DNA? You actually call it DNase. Adding ase just lets you know that it's an enzyme. Not every enzyme ends in ase, but most of them do. Now, here's the thing. If I have a beaker of sucrose and I just let it sit out, sucrose will break down by itself, right? Sucrose will spontaneously break down to glucose and fructose without you doing anything to it, right? But it's a very slow reaction. You might get one out of the whole beaker that breaks down every day, all right? It's very, very, very slow. So if it happens by itself, slow or fast, if it happens by itself, what's the delta G? The delta G is still negative, it's still spontaneous, but it happens so slow that it's just too slow. Life goes faster than that. So what enzymes do is they speed up reactions that would happen anyway. It just makes them faster. All right. So what sucrose might do, it might take sucrose and take it into its active site and like pull on this bond right here to encourage the bond to break on its own. It applies pressure, right? This diagram, this is an important graph. It's called the activation energy. So here's my reactants. Say I have A and B, C and D, or say molecule AB, molecule CD, and I want to turn it into molecule AC and molecule BD. See how the bonds got shifted? Just looking at this graph, is this reaction spontaneous? The answer is yes, because the delta G is negative. That little dot should be a less than sign. These dots don't convert, right? I'm not quite sure why, but that should be a less than sign, right? So this reaction happens. It happens for free. You ain't gotta do anything to it, but it happens very, very slowly because there's this bump called the transition state. And this diagram kind of shows the transition state. It's having this little wobbly lines because you're putting pressure on the bonds. So to get the reaction to happen, you do have to put some energy into it to get it over that hill. So overall, is the delta G still negative? Yes, because compare the reactants to the products, it's still a negative delta G, but you got to push it up the hill to get it there. What enzymes do is they lower this E sub A. They lower the activation energy to make it easier to go over the hump. Enzymes do not change the reaction, the reactant's energy. Does not change the energy of the products. It only changes the energy of the transition state. This diagram shows it. The reaction without the enzyme looks like that. The reaction with the enzyme made it a little bit easier. All right. So common multiple choice question: what do enzymes affect? All right. If it's multiple choice, reactants no, products no, delta G no. It changes the activation energy, and that's it. All right. This point here, enzymes do not make non-spontaneous things spontaneous. If it's not spontaneous, an enzyme really isn't going to help you. Instead, they speed up reactions that would occur on their own and just makes them faster. All right. Notice, like, you don't have to have ATP to make this happen. You just need to have the enzyme. Things that are not spontaneous requires ATP. This reaction does not require ATP. Some more things about enzymes. So the enzyme is the protein that does the reaction. The substrate is the reactant, is what the enzyme acts upon. So what's the substrate for sucrase? Sucrose, right? The enzyme substrate complex. So just look at the picture. Here I have an enzyme. The enzyme's a big globular molecule, but there's a site called the active site. That's where the substrate, or it could be multiple substrates, where it fits. If the active site got blocked, the enzyme wouldn't work, right? Because the active site has to be available for the substrate. And notice if this enzyme's shape got changed, if the enzyme kind of unfolded and the active site had a different shape, it wouldn't work either. That would be denaturing the enzyme. Enzymes usually work on a single substrate. So if you want to break down sucrose, you need sucrase. And sucrase really only works on sucrose. There's exceptions to that, but generally speaking, one enzyme works on one substrate. We already said this, the way enzymes work, they lower the activation energy. They could either, like, this is showing an enzyme that brings two substrates together and forms them. Could these two randomly come together all on their own? They could, it'd be random. The enzyme might have bonds that help attract the two substrates. Maybe they're ionic bonds or hydrophobic interactions, which brings the substrates together, which gives them encouragement to bond. I can't talk today. Or it could do the opposite and strain substrate bonds to encourage them to break. This just shows the same thing just as a cycle. Two substrates come in, the enzyme puts them in the right place, the reaction happens, you kick off the products, that's done. And then the enzyme, guess what it can do? It can go do this again and again and again and again and again. All right, let's wrap it up. So factors that can affect enzyme activity. So temperature. In chemistry, usually heating a reaction makes it go faster. And that can happen for biology too. But if you heat an enzyme too hot, it denatures, right? This graph shows me, typical human enzymes might, 37 degrees Celsius is about human body temperature. If I heat it up too hot, you denature the enzyme. I mean, if all your enzymes denature, you're dead, right? This is showing the curve for heat-tolerant bacteria that might live in like a hot spring that like it hot. They can tolerate much higher temperatures. So heating an enzyme might speed it up to a point, but heating the enzyme too much, you're going to denature the enzyme, which obviously is bad. Same thing with pH. Enzymes have optimal pHs. Your stomach might like it pH 2. Your intestines might like it pH 8. Your blood might like it pH 7.6. If the enzyme is out of its optimal pH, it doesn't work right because it denatures. Remember, pH is H pluses and OH minuses, which those can interact. Remember how we said, you change the pH of a molecule, you might change its structure? I think we did this back in chapter 3. If you change the structure of the enzyme, it's not going to work right because the enzyme structure determines its function. Form fits function. The structure determines the function, right? Salt concentration, if you add ions, those ionic interactions can mess with the active site. Cofactors are just they're non-protein enzyme helpers. Things like vitamins or metal ions are molecules. They're not the enzyme itself, but they might stabilize the enzyme's active conformation, so they're helpful to have. Not having the right vitamins means the enzymes might not work properly. What would happen to a reaction? So say I have a reaction that's going fine. What happens if I add more enzyme to the reaction? Would adding more enzyme to the reaction speed up the reaction? It would, to a point, right? Once all the substrate is used up, if I've converted all the substrate to product, adding more enzyme, the reaction's done. What if I did that and I added more enzyme and the reaction, I converted all the substrate to product? What if I then added more substrate? Would that increase the reaction rate? Yes, it would, all right? Because if you add more substrate, there's more things for the enzyme to act upon. Enzyme inhibitors, so look at the diagram. Here I have this nice purple enzyme. Here's the substrate, binds great. A competitive inhibitor, and that E got shifted to the bottom. Competitive inhibitors are molecules that compete for the active site with the substrate. They get in the way. A competitive inhibitor blocks the active site. That's bad, right? A non-competitive inhibitor binds somewhere else on the enzyme. See, I have this orange molecule binding elsewhere. It changes the shape of the active site, which is still bad, but it's binding somewhere else. Different toxins, poisons, pesticides, antibiotics tend to be molecules that inhibit enzymes. If you inhibit too many enzymes, the organism is going to die. If I inhibit enzymes that work in your nerves and all your nerves stop working, you're dead. Those are what neurotoxins are. So let me ask you a question. So say I had an enzyme and say that I added a competitive inhibitor, all right? The competitive inhibitor fights for the substrate with the active site. What would happen to the reaction rate if I added more substrate, all right? Adding more substrate would compete with the inhibitor more, and that would cause the reaction rate to go up. So if this is my problem right here, and I add more substrate, like say I have 10 substrates and 10 inhibitors, there's a 50-50 shot. Say I have 10 inhibitors and 1,000,000 substrates. Well, the inhibitor has hardly any effect because the substrate competes with it more, all right? Now, what if I have this going on, right? And I add more substrate, that's not gonna overcome the problem, right? So adding more substrate helps fight competitive inhibition. Adding more substrate does not help fight non-competitive inhibition. Say that I had 100 enzymes and I had 100 inhibitors. All the enzymes are inhibited. If I add more substrate, it ain't gonna make a difference because all the enzymes are used up. What if I have 100 enzymes, 100 inhibitors, and I add 100 more enzymes? Will that overcome the problem? Yes, it would, right? So here, adding more enzyme overcomes the inhibition. Here, adding more substrate overcomes the inhibition, all right? Okay, good. wrapping it up, allosteric regulation. See, I have this enzyme. Let's just look at the diagrams. It has 4 subunits. There's 4 active sites. Like this is an active site. This little green thing is an activator. It bonded to the molecule at somewhere other than the active site, but it stabilized the active site. This is an inhibitor that bonds somewhere else in the active site, but it stabilized the sort of non-functional version. Allo means other, steric means site. So allosteric regulation involves a site other than the active site. So adding more substrate would not make a difference, but it either stabilizes the active conformation or stabilizes the inactive conformation, and both can happen. Cooperativity, this shows a similar idea where I have 4 active sites. This one does involve a substrate. Here I have a substrate that bound to 1 active site, and it stabilized the other 3 active sites. So if the substrate helps stabilize the active conformation, that's called cooperativity. Hemoglobin is an example of a molecule that does this. Hemoglobin, which isn't an enzyme, hemoglobin is a protein in your blood that carries oxygen. But hemoglobin has 4 binding sites for oxygen. And if you have 1 oxygen bound, it encourages another one, then another one, then another one. If you have none, it's like the least effective version of hemoglobin. And last thing, feedback inhibition. Cells don't like to be wasteful. Cells don't like to make things that they already have. So this is a situation where the product that you made turns off the pathway. So let's look at this diagram. This is just the same diagram, just bigger. This is an enzyme pathway that is going to be making the amino acid isoleucine, and you begin with the amino acid threonine. So if, say you just had a meal and you just ate lots of isoleucine, like you have it in plentiful numbers, do you need to make it? No, you don't. So isoleucine is actually an inhibitor of the enzyme in this first step, right? The enzyme is threonine deaminase, right? So the product turned off an earlier stage. Now, all your isoleucine goes away. What happens then? Well, if it all went away, you need to make it again, right? If all went away, your inhibitor's gone. The active version of the enzyme is the one that's most predominant, right? So this is a way of a cell regulating resources. Cells don't want to be wasteful. They don't want to be wasting their limited number of resources. So the end product inhibiting an earlier step is feedback inhibition. If you don't need to make it, the cell doesn't want to make it. Okay, that was good. Hope that was helpful. I will see you guys next time.